Hello, I'm Peter Ney. I'm a postdoc at the University of Washington, and today I'm going to talk about work we did looking at DNA sequencing flow cells and the security of the molecular digital interface. Now, before getting into describing the details of our work, I'm going to start with a quick overview of how DNA sequencing works. DNA sequencing is the biochemical process used to read the information that's encoded in physical DNA molecules and convert that information into a digital form. In practice, this means determining the linear sequence of DNA bases, also known as A, C, G, and T, that make up each of the DNA molecules within a sample. In recent years, modern DNA sequencing technology has improved enough so that high-throughput DNA sequencers are capable of sequencing hundreds of millions of DNA molecules within a sample at the same time, which enables users to sequence up to multiple human genomes at the same time. And this has led to an exponential increase in the amount of DNA sequencing that's done. And because of this, DNA sequencing has become important to a number of domains, including medicine for medical diagnosis, genomics for research, forensics for sample identification, and novel applications like familial searches, which allows users to try to identify close relatives just using genetic information. However, there are a number of applications in the DNA sequencing world that leverage DNA and DNA sequencing for what would typically be associated as digital purposes. And probably the most well-known example of this is a field called DNA data storage, which tries to use synthetic DNA to both write and read information from DNA molecules. And the reason you might do this is because DNA is highly stable and can last for hundreds of years. And because DNA is the information carrier for life, we can assume that we're going to have technology that's capable of reading DNA for a long time. So when viewed abstractly, you can actually think of a DNA sequencer as something like an interface which straddles the molecular and digital realm. And this is important because this means that issues or problems or design decisions that take place in the molecular realm can actually influence and impact the digital world. So all of this got us interested in understanding the security and privacy implications of a specific piece of hardware within a sequencer called a sequencing flow cell. And the flow cell is important because it's actually the site of the molecular digital conversion and is where sequencing takes place. Right now, there's two main classes of sequencing flow cells that are used in practice, one made by Illumina and the other by Oxford Nanopore. In this work, we focus on the flow cells created by Illumina, which use a sequencing by synthesis type sequencing technology. Sequencing by synthesis works as follows. First, a DNA sample is pre-prepared in the wet lab to make it ready for sequencing. This usually results in each of the molecules in the sample being somewhere in the order of 100 to 300 bases in length. Next, these short DNA molecules are washed over a flow cell surface, which is sticky so that the DNA molecules bind to the flow cell surface, where each of them can then be individually sequenced by detecting different fluorescent signatures for each of the bases that are being added. These signatures are picked up by high-resolution cameras, which can then be used to reconstruct the sequence of each of the separate DNA molecules. This then results in a large DNA sequencing file containing the sequence of each of the strands, one after the other, usually hundreds of millions of strands per sequencing file. Because DNA sequencing flow cells are so integral to the sequencing process, we wanted to ask whether flow cell design and architecture could have security and privacy implications. In particular, we thought they were interesting to study because unlike a lot of computational platforms, they actually process molecules as opposed to being more of an electronic variety. And molecules have interesting properties. So for example, they can diffuse, react, and can be stable for long periods of time, which all might have implications for security and privacy. In particular, we were interested in understanding two issues. The first was whether DNA left on a flow cell could act somewhat like a hard drive with data that wasn't properly deleted. And so, for example, could this lead to something like data remnants issue? And the second issue, as I'll describe later in the talk, is that sequencing is actually done in parallel with multiple samples to increase throughput, which raises issues of parallelism and concurrency and possibilities of multi-tenancy problems, similar to what we've seen in things like cloud environments. Data remnants is a well-known privacy issue from classical hard drives, both hard disks and solid-state drives, and it results when digital information is encoded in a storage medium, and then when that file is deleted, it isn't actually properly deleted because there's a remnant of it left on the physical storage media, which can then be recovered using forensics techniques. And similarly, we wondered whether there was something analogous that was happening with DNA sequencing flow cells. So for example, if you were to sequence medical information or a human genome sample and were to dispose of the flow cell using standard disposal procedures, could you actually recover the DNA that was on that flow cell, resequence it, and recover most of the usable information that was from the original sample? DNA sequencing flow cells are designed to be single use. And so our first question was whether it's possible to extract usable DNA information from a flow cell that was used in a prior sequencing run. And we thought this might be possible for a few reasons. First, DNA is sticky, and we thought there was a good chance that some DNA would remain bound to the flow cell surface after sequencing was finished. Second, DNA is stable at room temperature and can last for months or years at a time, still containing usable information. Third, even low yields of DNA may be recoverable because DNA can be enzymatically amplified to increase its yield. With this in mind, we were able to develop a washing procedure which enabled us to collect residual DNA left on a flow cell through a number of flushes. We found that we were able to flush a flow cell 5 times with fluid and still collect a usable yield of DNA. And in particular, this residual DNA could be easily amplified due to the design of the DNA molecules that was specified in the pre-sequencing wet lab process. Therefore, we had a simple method to both collect residual DNA and increase its yield with enzymatic amplification. And while the previous experiment showed that we could collect some residual quantities of DNA, it wasn't clear whether this DNA actually contained any useful information. And so to test this, we ran 2 different sequencing experiments with an Illumina sequencer, trying to understand whether we could recover information in a high redundancy environment, one with lots of error correction, in this case, a DNA data storage file with a number of images, and secondly, more of a medical context or a genomics context where we actually wanted to sequence a human genome sample and see what fraction of that genome we could recover. In the first experiment, we tried to recover DNA from a used flow cell where the DNA originated from a sample that encoded a number of digital images. This allowed us to understand how residual DNA recovery might work from a sample with a high level of redundancy, which is typical in a DNA data storage file. And in fact, we were able to fully recover the images from the DNA left on a used flow cell. More interestingly, there were significantly more substitution rates, which are flips between bases, in this case about 6 times higher in the residual sequencing run than the original run. We suspect largely due to the additional amplification of the DNA we did as part of our recovery procedure. However, we did notice that there were similar error rates of other kinds of errors. And in particular, they were still well within the error correction budget that was allowed by the DNA data storage decoding process. Next, we wanted to understand how data remanence issues might affect a more natural or biological DNA sample with much lower levels of redundancy. And when we repeated the same process of trying to recover a human genome sample from a used flow cell, we found that in fact, yes, we did recover significantly less of that sample than the original run. However, there was still a substantial amount of unique data that we were able to pull from it. In particular, we were able to recover about 1.8 million strands from the second residual sequencing run compared to 4.4 million in the original. Interestingly, around half of that DNA was never actually seen in the original sample, implying that we're actually pulling new DNA that was not sequenced in the original sample when we do our washing procedure. However, in terms of actual usable information, this was a significant amount of recovery because there is some amount of duplication that naturally occurs in a typical medical or genomic sequencing process. And so in general, you expect around 30x coverage, which means on average, each base has about 30 DNA molecules, which encodes that base. And in our recovery procedure, we were getting more on the order of 10 to 15x recovery. So it's not quite equal to the original run, but it's still a significant amount and an amount that would allow you to determine most of the interesting genetic information in a sample. So in summary, when looking at data remanence issues on flow cells, we were able to develop a recovery procedure that allowed us to collect and amplify DNA such that we could recover most of the DNA in a DNA data storage or high redundancy context, as well as in a lower redundancy context like medicine and human genomics. But in both cases, it was sufficient for a significant amount of recovery, raising security and privacy issues for flow cell disposal. In the paper, we discuss a number of defenses and solutions to this problem. Please see that for more details. The second question we had with regards to flow cells and security and privacy was how issues of parallelism and multi-tenancy could affect security and privacy. In particular, we were interested in studying the security and privacy implications of a multiplexing procedure that's done often in high throughput sequencing environments. And this process allows up to 96 separate unique DNA samples to be concurrently sequenced on the sequencer on the same flow cell at the same time. And then later, the data from that sequencing run is demultiplexed into data for each of these individual samples. And this is done using what are called chemical barcodes, which are enzymatically added to each of the DNA molecules in the sample before sequencing. So this occurs during the pre-sequencing procedure. And what happens is each sample is given a unique barcode so that when that molecule is sequenced, the barcode is read and can then be used to demultiplex that DNA strand to the correct file. The issue is that this demultiplexing procedure has a small but predictable amount of error. And this error is referred to as barcode crosstalk, and it's caused by both chemical issues on the flow cell as well as the way the flow cells are actually designed and the way that DNA is flowed over the flow cell surface. And what happens is that small numbers of reads, somewhere in the order of 0.0001% to 0.1%, are actually assigned to the wrong file. And so in a typical sequencing run, this could be somewhere on the order of 200 to 200,000 DNA reads. Now, previous work has looked at this crosstalk phenomena and found that it was possible to use crosstalk to predictably read information or small amounts of information from other DNA samples. But in this work, we were actually interested in understanding the reverse of that process, which is, could one malicious sample use crosstalk to actually write information into other data files? And in particular, trying to understand what the security implications could be of these unauthorized writes. And in general, these risks are going to be specific to the downstream processing and analysis that's done with that sequencing data. In this work, we were interested in studying a specific kind of analysis called variant calling. And variant calling is a very common genetic technique to try to identify differences in an individual genomic sample. So in this case, the goal is to identify markers in a genomic sample which differ from the standard reference sequence. And recall that in a human sample, humans have 23 chromosomes. These chromosomes come in pairs. Therefore, you actually are determining pairs of markers at each position. Recall that DNA sequencers are designed to only sequence short DNA molecules. And so what's actually done when sequencing a genomic sample is those long chromosomes, which are tens of thousands of bases long, are broken up into small pieces. Each sequence is then aligned to a reference sequence using standard string alignment, and then those markers of interest are evaluated statistically to see whether you would have the genotype that you would expect. So for example, in this case, if at marker 1, you have a T, however, all of the reads which align to that sequence from that genome sample have an A, then you would expect that that genotype is 2 A's at that marker. Similarly, if you have a G for that reference sequence, and you were to do string alignment against that reference, all of those DNA reads, if half are G and half are A, you would predict that the genotype at that marker for that sample is an AG. Now, coming back to our original question as to whether you could use an unauthorized write in a malicious manner, we wanted to ask the question as to whether it was possible to write a sufficient number of molecules into another sample that would then effectively manipulate the variant calling algorithm. So in this case, imagine that you have a normal human genome sample, which has 2 A's at a particular marker, and if you were to write a bunch of strands with a T at that marker, but were otherwise the same, the question is, would that be enough to modify the variant call that would be called at that marker? And in this case, maybe flipping it from homozygous AA to an AT, or even a TT. And so what would happen if you did this, if you were able to write a sufficient number of DNA molecules containing a T, what you would expect is all of these would then align to the same position, except in this case, the variant caller would statistically infer that, yes, maybe this is actually 50% A, 50% T. Therefore, we would expect that they are an AT at that location. All right, with this in mind, we then went ahead and tried to design an experiment which then tested this hypothesis. And so what we did was we started with 2 samples. The first sample was a standard human DNA sample like before, and the second was a malicious sample which contained just synthetic DNA that was short, 400 bases in length, that was designed to look like a specific locus that we were targeting. And the question was, could we actually alter that marker in the human data, even though those samples are supposed to remain independent? And in particular, what we tried to do was design synthetic molecules that looked like the marker which is responsible for sickle cell disease, which is a single base substitution in the beta globin gene. And so, in effect, the human sample, which normally is AA, which is a sign for being normal or not having sickle cell disease, and change that person's variant calling status to either being a carrier, which is AT, or disease status, which is TT. So we went ahead and ran this experiment and found that there was, in fact, enough crosstalk observed, a very small amount in this case, around 600 reads, which was sufficient to manipulate the variant in the normal human DNA sample. And in particular, we were able to alter the genetic prediction for that sample from normal, AA, to AT, which is signifies that that individual will be a sickle cell disease carrier. So to conclude, I'm going to discuss why I think this work has relevance for the computer security and privacy community. And I think the first reason is that it's important to understand that state-of-the-art biological instruments, like DNA sequencers we studied in this work, are actually becoming more computerized than ever before. And this is starting to raise new computer security and privacy issues that haven't been previously considered. And in particular, in this case, the vulnerabilities we uncovered were caused by complex interactions that involved molecular chemistry, flow cell design, and downstream data processing. And this is important because it means that reasoning about security and privacy is going to be hard and interdisciplinary. And lastly, I just say that we believe that improving security and privacy in the wet lab is going to continue to be a very important area to study, especially given its high impact. And with that, I'd like to stop and ask for any questions. Thank you for listening.